Alright, welcome everyone to our video today discussing the mechanisms, biology and some pathology of RNA splicing. RNA splicing is a cellular process within the central dogma in which the intron coding regions of the DNA are excised from the mRNA sequence to leave the final exonic sequence that will determine the polypeptide sequence. Introns have arisen in all eukaryotes and don't code for the protein structure, so have to be removed via the process of splicing. Splicing has to have high fidelity as incorrect ligation of exons can result in frame shifts and the polypeptide sequence consequently forming a non-functional protein. The process of RNA splicing is a crucial step in the maturation of pre-mRNA into functional mRNA. It's involved in the removal of introns, non-coding regions, and the precise joining of exons, or coding regions, to ensure the accurate translation of genetic information. The splicing mechanism is orchestrated by a large dynamic ribonuclein protein complex known as the spliceosome. The spliceosome comprises small nuclear riboprotein particles, or SNRPs, and additional proteins, and its assembly is ATP dependent. Splicing occurs at certain sequences called splice sites that are present at the exon-intron junction that signals the spliceosome where to cut and ligate RNA. The consensus sequences forming the splice sites follow a GUAG rule to mark the boundaries between the introns and exons, which can be recognized by the spliceosome. They are conserved across eukaryotic organisms, highlighting the essential and evolutionarily conserved nature of splicing mechanisms. The SNRPs act as ribozymes and undertake RNA-catalyzed splicing reactions, specifically facilitated by the U2 and U6 snRNAs, which recognize the nucleotide sequences marking the intron boundaries. They also bind to cause the folding of the mRNA at these intron boundaries for the alignment of mRNA to bring the intron-exon boundaries together for their cleavage. Introns are excised by an intricate two-step nucleophilic process. During the splicing process, the 5' end of the intron is cleaved and a looped structure termed a lariat is formed, involving a 2' to 5' bond between the G at the 5' end of the intron and the branch point. Subsequently, the 3' end of the 5' exon attacks the 3' end of the intron, leading to the ligation of exons and the release of the intron as this lariat structure, which is eventually degraded. This overall process is ATP dependent, as the number of phosphodiester bonds is conserved. This process is ATP independent, as the number of phosphodiester bonds is conserved. Proteins associate with the mRNA near the exon-exon junctions, forming the exon junction complex. The exon junction complex plays a role in mRNA export, translation regulation, and nonsense-mediated mRNA decay, which play an important role in degradation, influencing mRNA turnover. Alternative splicing is a crucial cellular mechanism that revolves around the different ways mRNA can be spliced and diversifies the coding capacity of genes by generating different mRNA isoforms, which in turn give rise to distinct polypeptides. This process significantly contributes to the regulation of gene expression, allowing for cell type or developmental stage-specific adjustments. Alternative splicing can occur through various modes, such as the use of alternative 5' splice sites with a common 3' splice site, exon skipping, or the utilization of a common 5' splice site with alternative 3' splice sites. These alternative splicing events can introduce variations in the final mRNA sequence, leading to the production of multiple protein isoforms with different functions. Aberrations in RNA splicing play a pivotal role in the pathology of various diseases. Aberrations in RNA splicing play a Aberrations in RNA splicing play a pivotal role in the pathology of various diseases, exemplified by the Wilms tumor suppressor gene, WT1. This gene undergoes extensive alternative splicing, generating over 20 isoforms that contribute to its diverse functions. Mutations in WT1 have been associated with several disorders, including childhood kidney cancer or Wilms tumor, severe kidney diseases, and gonadal dysgenesis, leading to male-female sex reversal. The genetic abnormalities underlying this tumor, often unknown, involve mutations in the protein coding region or introns, disrupting the normal splicing pattern. Such mutations result in truncated forms of the WT1 protein, lacking essential domains, particularly its DNA binding domain. Consequently, these truncated forms lose their function as tumor suppressor transcription factors, contributing to the development of a rare developmental defect affecting the kidney and gonads. The intricate interplay of going back to the end of the sentence where it ends with DNA binding domain. Consequently, these truncated forms lose their function as tumor suppressor transcription factors, contributing to their development of Wilms tumor. Furthermore, WT1 mutations have been implicated in Fraser syndrome as well, a rare developmental defect affecting the kidney and gonads. The intricate interplay of RNA splicing variants in Fraser syndrome underscores the nuanced impact of splicing abnormalities on disease manifestation, with the plus KTS and minus KTS isoforms of the polypeptide sequence influencing the functionality of the WT1 protein in distinct ways. In summary, RNA splicing, orchestrated by the spliceosome and influenced by various protein splicing factors, is a pivotal cellular process essential for precise gene expression and